Hello students, so today we are going over the study guide to best prepare you for your exam, which is tomorrow. So please watch this video to best prepare you for your exam. Here is the picture of your study guide, and it says you need to know the 9 abdominal regions and the cell structures diagrams. So here is a copy of your 9 abdominal regions, which you can also find in your Chapter 1 slideshow. We want to know these 9 regions right here and be able to state them on your exam. And for your own benefit, I went ahead and did this practice cell diagram addition to your Unit 1 module of the body org, so you should be able to click on here, right here for the practice in order to best prepare you for the exam. So that took care of our 9 abdominal regions and our cell structures diagrams. Okay, vocab. We want to be able to define all of these words: anatomy, physiology, homeostasis, concentration gradient, and cell differentiation. So for anatomy, it is the scientific study of the body structures or form, and this is how we can determine where a structure of the body is. Physiology is the study of the chemical and physics of the body, which describes how things are working. So it answers the question how. Physiology answers how, and it describes the function of structures. Anatomy answers where, which is where structures are in the body. That took care of here our anatomy and physiology. Homeostasis is going to be, the definition is to maintain an internal steady state of the body, and it helps do this by negative and positive feedback. There are 3 components of homeostatic systems, which are receptors, control centers, and effectors. There's a huge video on homeostasis in your course lectures, so if you need a review of homeostasis, please go back and watch that lecture. Okay, let's talk again about concentration gradients. Concentration gradients was commonly missed on the study guide, so concentration gradients is having 2 different environments with varying concentrations of solutes. So a concentration gradient just basically means there's 2 environments, and one environment has a different concentration than the other, which creates a varied or 2 different environments with 2 different concentrations. And for concentration gradients, we also want to understand simple diffusion, which is where the movement of solutes occurs from areas of high concentration to areas of low concentration, whereas osmosis is the movement of water from areas of low solute concentration to areas of high solute concentration. So water is going to want to move in a direction that is going to lower the concentration of that environment, whereas solutes are going to want to also move from areas of high concentration to low concentration, thereby diluting the environment as well. Cell differentiation occurs through different processes where our cells differentiate to change in order to carry out their specific functions. Okay, and for our medical terms, cyte means cell, hist means tissue, hyper means above, hypo means below, eryther means red. Honestly, we don't need to know this one, so ignore it. Hemat means blood. Leuc means white. Iso means same. Superior means above. Inferior means below. Cranial means towards the head. Caudal means towards the tail. Posterior is the same as dorsal, and it means on the back side of an anatomically correct position. Ventral or anterior means front, and it is the front of the body talking about our anatomically correct position. And for our anatomically correct position, we want to remember that we have our friend here looking like this, where the palms are facing the front and the back of our hand would be facing the back, posterior. This would be our nose. So this would be the anterior view of our anatomically correct figure. And when we're speaking of body parts, make sure you're always talking about it being an anatomically correct position, including that, here's our guy again. That would be incorrect, right? Our hands are facing down in our anatomically correct position. And we also want to remember that we're talking about his right or left side. So this is his right side of the body, and this is his left side of the body. Make sure that you guys understand that. Lateral would be towards the outside exterior part of the body. So when we're looking at our anatomical figure, this would be lateral and this would be medial when we're talking about half of the body. So when we're talking about the right half, this would be correct. And when we're talking about the left half, this would be correct. Lateral, medial. Towards the midline or away from the midline of the body, where this is our anatomical figure. For our proximal and distal, we're also, remember, talking about anatomically correct position. Our proximal would be towards the point of attachment, and our distal would be further from the point of attachment. And most of the time, we're talking about extremities here. So here's our guy. This point here would be proximal to this point here. And if we're talking about our legs, this point would be proximal and this point would be distal. When we're talking about deep and superficial, we're talking about close to the skin or deep to the organs. So deep would be deeper inside of the body, and superficial would be closer to the skin. And exocytosis versus endocytosis, we're not really too worried about that, but exocytosis is going to be excreting to an external structure, and endocytosis would be kind of secreting from an internal structure, but we're not going to be worried about that for the exam. All right, for the body systems, we are going to talk about these really quickly. There is a video published from your course modules that you guys should review. I've also went ahead and added that to, okay, I will add that here to where it's an external link, which should make it a little bit easier for you guys to view. But for now, we'll just go over these really quickly. We can remember the body systems with the mnemonic remind curls. Remind curls. All right, here's our remind curls, where this would be our respiratory or reproductive. So for our R's, we can put respiratory. And here we have reproductive. E is endocrine. M is muscular. I is integumentary. N is nervous. D is digestive. U is circulatory. U is urinary, which in that video is called the excretory. R is reproductive. L is lymphatic. And S is skeletal. Pardon my terrible handwriting. Okay, and the functions of each as laid out in that video, well, our reproductive is to reproduce offspring, and the important structures in that are our sex organs. Endocrine is for communication and aids us in our homeostatic feedback loops, and one of the important structures in that are hormones. Muscular system is for movement, and the important structures in that would be our muscles. We have skeletal muscle, smooth muscle, and we're going to talk more about that as the semester continues. Integumentary has the function of temperature regulation, and the important structure in that is our skin. Our nervous system has higher function and is our control center for our body, and it consists of the structures our brain and spinal cord. The digestive system has the function of absorption and digestion, and important structures of that include our intestines, stomach, mouth, and teeth, our anus, and our rectum as well. Our circulatory system has the function of transportation of nutrients and consists of our blood vessels, including arteries, veins, and capillaries. Our urinary system, also called the excretory system, and that fast osmosis video, has the function of excreting waste from our body and has the important structures of our kidneys, bladder, urethra. Then our respiratory system has the important function of intaking of nutrients, exportation of waste, and has the important structures of our lungs, bronchi, and our trachea. Our lymphatic system has the function of immunity. It has many different structures in here that we're not worried about, but we need to know the lymph nodes, thymus, and spleen are part of the lymphatic system. And finally, our skeletal system provides the framework of the body and aids in movement and has the structures of our bones. Okay, for number 5, compare and contrast the two types of feedback. What are the reasons these mechanisms are important? So feedback is going to assist us in maintaining that homeostasis in our body. For our negative feedback, we have the final effector is going to negate the original stimulus. That is the big point. So this is our example. We also talked about the regulation of glucose in the body, so please go back and review that video if you guys have any questions. Our body temperature is increased, okay? So then the nerve cells in our skin and our brain are able to sense this change in body temperature. This is going to send information to the regulatory center within the brain, the control center, and then our effector, such as our sweat glands throughout our body, which are located in our skin, part of the integumentary system, is going to sweat, which is going to allow the external environment to have conduction of, and it's basically going to decrease our body temperature. We're not too worried about how this happens. That's going to be considered the physiological reason why our skin is able to help us regulate our temperature. But for the first test, we're not too worried about that. We want to understand negative feedback. The effector is going to negate the stimulus. Here we are with our positive feedback. We have the stretch receptors in our uterus are going to increase stretch as labor begins in women. The head pushes against the cervix. The nerve impulses from the cervix transmit to the brain. These stretch receptors tell our brain, okay, there's more stretch. The brain stimulates pituitary gland to secrete oxytocin, which is a hormone. The oxytocin is carried to the bloodstream and has the receptors of the uterus that can bind to this oxytocin. And this complex of our receptor binding to oxytocin is going to stimulate increased uterine contractions and helps push the baby towards the cervix to help us carry out labor and delivery. So, what's the difference? Well, on positive feedback, our effector is going to basically amplify the stimulus. So due to this oxytocin receptor complex, we are going to increase more stretching of the uterus, and that stretching is going to therefore stimulate oxytocin to be increased again. Oxytocin's increase is going to increase stretch of the uterus, and therefore it's a continuous positive feedback loop to increase the original stimulus, which is the increase of stretch receptors causing the increase of oxytocin. And it goes on and on and on. All right, that would take care of our comparing and contrasting the two types of feedback. Why are these mechanisms important? Well, it helps us maintain homeostasis. Compare and contrast the ways in which concentrations of molecules can be varied throughout the body. Okay, so here I want you guys to focus on our body compartments. So we have the extracellular fluid which consists of our plasma and interstitial fluid, and our intracellular fluid, which consists of our intracellular fluid. All right, so here we talked about a lot in this video. Please go back and review that video from your Start Here page in your module if you are confused on what we're talking about right now. So in our extracellular fluid, like we said, we have plasma, which is within the blood vessels, and our interstitial fluid, which is between the cells. So here we have our blood vessels showing our intracellular space. Then between each cell, we have extracellular fluid, or interstitial space. And then within our cells, we have the intracellular environment, which is broken apart from the external environment by the phospholipid bilayer, or our plasma membrane. Okay, let's go look at our cell picture again really quickly, because our extracellular environment is based on the cell. So when we're looking at this picture, we should think of a whole bunch of cells lined up next to each other, and this right here breaks apart our cells by this phospholipid bilayer, which we're going to talk a little bit more about. What we want to know is that there are differing solute concentrations within the plasma, the extracellular space, and the intracellular space. And these solutes concentrations follow a few different rules of diffusion. So active transport and passive transport. Passive transport talks about diffusion, where solute concentrations move from high concentrations to low concentrations, separated by the phospholipid bilayer, having these two environments be separate from one another. When we're talking about active transport, this is where our solutes are going to move against their concentration gradients by the use of ATP and protein channels. Active transport, therefore, is going to move against concentration gradients, where our solutes can move from areas of low concentration to high concentration with the addition of ATP. Diffusion, on the other hand, is the movement of water, where water moves from areas of low solute concentration to areas of high solute concentration. We're going to let this animation restart so we can actually see what diffusion looks like. Right now it's going in the reverse direction of osmosis, but here we have osmosis, where our water is the moving part, and it's going to move from areas of low solute concentration to areas of high solute concentration when uninhibited and separated by a membrane, such as our phospholipid bilayer. Here we have salt molecules, which have a high concentration on the right side of the screen, and water is going to move from areas of low concentration to high concentration to try to dilute solutes. And we have the example of our tonicity here, talking about red blood cells, basically cells, and what would happen when we have hypertonic, hypotonic, or isotonic concentrations, which are all part of our medical terminologies for this chapter. So what we need to know is that when you put a cell in hypertonic solution, it's going to shrivel. Why does this happen? Well, hypertonic solution has high solute concentration, and our red blood cells have it, before you put them in solution, the biconcave shape. But when you put these cells into hypertonic solution, diffusion happens where water is going to leave the cells to dilute the concentration of the solution around the cells. So this behavior is demonstrating osmosis of water, where the water is going to move from the inside cellular environment or the intracellular environment to the extracellular environment in attempts to dilute the concentration of solutes within the solution. Isotonic refers to having the same concentration of the cells. So when you put red blood cells into isotonic solution, there's no net movement of water, because the tonicity of the water is equal to that of the cells. So water is neither going to go influx into the cell or efflux out of the cell. Finally, we have hypotonic solution, where the concentration of the solutes of the surrounding extracellular environment is less than that of the red blood cells. So when you put a red blood cell into hypotonic solution, the water is going to move or diffuse or undergo osmosis from the extracellular environment to the intracellular environment, causing these cells to swell up due to the influx of water via osmosis into the cell. Hopefully that makes sense. Here's another mnemonic here for you, where we can see the hypotonic, isotonic, and hypertonic solutions. What happens when we have a hypotonic solution? Our cells are going to swell. Isotonic, there's no net movement of water because the solute concentrations are equal. And hypertonic, our cells will shrivel due to the tonicity of the extracellular fluid.